So we have Denise Kirschner as our second talk, is going to be talking about her multi-scale modeling efforts on tuberculosis infection and treatment. And I'll turn it, I don't want to take any more of her time. I'll turn it over to her immediately.
Okay. Well, I really would just rather talk about James's talk because it was awesome and I have so many questions and thoughts. So I'm kind of frustrated. I tried to write everything down. I hope I remember, Jimmy, when we're done. But that was amazing. I wish I'd known you were talking about that because maybe I would have talked more about lymph node stuff. I'm not going to spend too much time talking about it today. So a lot of you have seen me give talks before on the biology and the study of tuberculosis. We've done it for about the past 20 years. We call it a systems biology approach because we believe systems biology is a use of multiple modalities to understand a single biological problem. And so we believe that that TB is very complex and it requires multiple modalities. So here I'm showing you a lung resection from a non-human primate with a granuloma in it. This is a human granuloma. Here's our computer simulation. So there's multiple actions of this. So TB is actually the #1 cause of death in the world due to infectious disease, and about 1.5 to 2 million people die a year. So even in the face of COVID, it's a major morbidity, and it certainly doesn't get as much attention because it tends to be a third world disease, but it is as important. And what got me interested in this disease was this trajectory that you either clear infection if you're exposed. Only a small percent of people get active disease where if they don't get treated, they'll die. But most people are able to contain it and experience what we think is latent disease. So what I'm showing you here on the right is a PET CT scan from a non-human primate where we've infected it with 24 bacteria that were barcoded. And you can see that each granuloma was formed by an individual bacterium. So these are collections of immune cells that serve to immunologically restrain and physically contain the bacteria because it can't really clear it. So it kind of builds a wall and walls it off. But what you notice here at 4 days 4 weeks post-infection, there's a single granuloma here, and at 5 weeks, it's spreading. So this monkey will probably do bad and die from the infection. And just for Jimmy's sake here, these are lymph nodes showing you that there is spread of multiple bacterial types and antigens to the lung-draining lymph nodes in this case, and I'll talk about that a little bit later. The problem with TB is that, of course, of these 90% that have been infected and are latent, there's a chance of reactivation about 5 to 10% per lifetime. And if you study viral infections or immunosuppression, things like HIV, that can reactivate tuberculosis and lead you to a 5 to 10% per year, which is a main problem. We have a fantastic animal model with a collaborator at University of Pittsburgh, Joanne Flynn, who we have a colony of 300 monkeys that provides much of the data that we use to calibrate and validate our models. So this is a multi-scale modeling consortium. So we believe that this is a multi-scale process, and the molecular scale dynamics, including drugs and chemokines and cytokines, are behaving, interacting with things at the cellular scale, coming together at the tissue scale to form these granulomas. And of course, there's organ scale dynamics happening with things moving in and out from the lymph node, as well as the blood compartment as things travel back and forth. And so this, of course, is contained within a whole host. So we care very much about building an agent-based model that can track every individual immune cell in lung tissue and what it's doing that leads to these granulomas, because they've been a veritable black box to study, similar to lymph nodes. And we've done similar studies like this for lymph nodes as well. And so we track immune cells of different types and bacteria, and things are diffusing. We also have vascular sources in our lung, and what's amazing about this is when we run this agent-based model, the emergent behavior is actually the formation of a granuloma. And this model has been continuously curated now for over 15 years with data and updated with new immunology and biology data as it becomes available. So here's an example from a non-human primate of an immunohistochemistry stained tissue showing a granuloma. And you can see here we have some dead tissue in the middle, surrounded by rings of, the blue is actually neutrophils. Then we have macrophages and then surrounded by lymphocytes. And so you can see that our simulation gives you a very similar type of a structure. But more importantly, we can track over time from the origin of dropping that initial bacterium into the system, how this thing dynamically changes. And as modelers, of course, our goal is to be able to manipulate this tool once we're sure it's working well to kind of make many predictions about what's going on. We use this model to study lots of different aspects of both immunology as well as things like vaccines and treatment. This granuloma is going to run for 200 days because infection with TB lasts a lifetime of a host, and these granulomas last a lifetime. We find tubercles in Egyptian mummies, so this is an ancient disease and that lives the length of the host. You can see here this granuloma that's forming is kind of splitting into two granuloma, and there's caseum developing in the center of both of them. So lots of bacteria present there. I'm just showing you here that our vast number of simulations matches what's identified in the non-human primate, at least through the first about 120 days. We haven't really had monkeys that are longer than about 4 months. We usually necropsy by then. So we use these analysis tools of uncertainty and sensitivity analysis to figure out how much parameter variation influences the model outcomes, and then if there are variations, which parameters are driving that behavior. And that's what the sensitivity analysis tells us. And it's really exciting because it can quantify the relationship over time. So what's contributing to the granuloma formation dynamics early versus what's happening later. And we published this paper many years ago now, and we have the programs available in MATLAB and R on our website for anybody to use that wants to perform these studies. So for this basic GranSim model that I just showed you, here is the result of the sensitivity analysis. So I'm showing you partial rank correlations on the right. It's similar to regular correlations, although these are for nonlinear systems. And you can see that the parameters that are playing a role early in infection in the first 50 days, very different from the parameters that are playing a role later in infection, out to day 200 here. And so we're able to sort of classify these in different categories, things that are involved with this tumor necrosis factor, rates that T cells are recruited to move around the lymph node, macrophage activation rates, how well the intracellular bacteria are growing, and what's going on with this dead tissue in the center of these granulomas. So this allows us then to have targets to do further study, build molecular scale level models that are submodels that we can put on the bottom of this mesoscale model to ask more questions, as well as build larger scale models to link to this. So we've spent a lot of work in this area, and I'd be happy to talk to anybody about those sorts of models that we've done. So now in this particular case, I just want to talk to you about why TB is so hard to treat. So there's three main reasons. The first is that the bacteria, it grows very slowly. For example, E. coli doubles every 20 minutes. It takes Mycobacterium about 24 to 48 hours to double. Also, there's antibiotic resistance. In terms of the granulomas, they themselves form a physiological barrier to antibiotic diffusion. So here's from a rabbit model, which form beautiful granulomas similar to humans. We've treated with two separate drugs, and you can see in this drug, it doesn't penetrate at all into the caseous area. In this drug, it can somewhat penetrate, but most of the drug is concentrated sort of here in the macrophage ring, not much in the T cell area here. And then it's patient unfriendly. You have to treat as long as 9 months. Most people are treated 6 to 9 months. Multiple drugs are given. The standard regimen is made up of isoniazid, rifampicin, pyrazinamide, and ethambutol, and there are many side effects. Now, most people don't normally finish their 10-day course of antibiotics, so actually completing 9 months is a difficult task, and non-compliance immediately leads to the emergence of drug resistance, so it's an incredibly important problem. The World Health Organization has now put a Stop TB campaign to the year 2030 out, and there is a lot of effort to shorten the amount of time that drugs are needed and maybe even reduce the number of drugs given to help compliance strengthen. So what we've done is taken this GranSim granuloma simulator model, and we've put a plasma pharmacokinetic and a tissue pharmacodynamic model running inside of it. And so another multiscale here for these molecules and tracking multiple transit compartments of drugs. So, first you have to swallow it, right? And then it has to get into your, different tissues, get to the plasma, then get into the periphery so it can get to the lung tissue. And then, of course, what are the dynamics? And what we assume here is that the antibiotic killing rate is proportional to the C50 or the half max of killing here. And this assumption is well known in the literature. So here's an example. Here's an example showing you that the drugs actually stay below what you need the C50 to be for most of the time. So these are different populations of bacteria because we look at the environment that the bacteria in that determines its growth rate. And you can see no matter what region it's in, the drugs are not above the C50 very long. And this is treating every day. So you can imagine why someone has to treat for 9 months to be able to really get at all the bacterial load. Something extremely interesting, here is an example granuloma, and I'm treating with 2 separate drugs. And what you can see here is when you treat with one drug, immediately the drug, this one here, isoniazid, has the half-life, it's basically gone. Rifampin is stayed around a little while before the second drug delivery is given on the next day, but you can see most of the drug is located outside of the granuloma and not penetrating into the granuloma. And so again, this is information that's really been a black box for pharmacologists, biologists studying granulomas, and we're able to visualize that here and make predictions about how we can improve this process. So one of the things we did is we were able to look at a couple of different types of granulomas. And we looked at the combination therapy that's given basically to everyone in the clinic right now. And it's given to you whether you have latent TB or active disease. You are given this therapy of HRZ unless you have a resistance, and then they swap in other drugs, which I'll talk about in a minute. So if your granulomas are all good, so it probably looks more like you're a latent person, you can see that if you give any of the individual drugs, you don't do well unless you've been given rifampicin, which is almost as good as given all 4 at the same time. But if you have granulomas that are sort of doing poorly and really probably likely in a person who's active disease, what you see happening is that only the combination will get you to all the granulomas being sterilized, which is what the goal is of treatment in this case, is to clear the bacterial load. But all of it takes at least 200 days here on treatment to even begin to get to that point in the bad granuloma case, and it takes about 120 days to reach that in the good granuloma case. Okay, so another major question is, well, what is the best antibiotic regimens? So what if you had, for example, 2 treatment segments, like an initial phase and a longer phase? What if you had 10 possible drugs and you had 4 drugs you were going to give per segment, and you had 5 possible doses that you could give for every one of the drugs, and you had, a frequency of once a day, twice a week, 3 times a week, 7 possible frequencies. That number of combinations is 10 to the 17th. And so we refer to that as the regimen design space. That's even too many for computational modeling to be effective. So what we have to do, and certainly too many to do clinically, is create an optimization system to study this. And that's just what we've done. So we sort of looked using 2 different methods, a genetic algorithm method and something known as surrogate assisted optimization, which comes from the engineering side of things. And so 2 things are important here. We're trying to come up with an optimization approach and also an objective function. So of course, genetic algorithms act in a way that you initialize a population, you see how it evolves over time, you have a stopping criteria, and you're done when you feel like you've hit the perfect fitness. Surrogate assisted optimization, you don't really have any idea what you're looking for, but you guess. And then you notice that you're looking for lots of local minima here for your or maxima, depending on what your question is. So we need an objective function for that, and here we've just used something simple like time to clearance and dose frequency, and we're trying to minimize that. And so the idea is you actually are creating a surface for the different things you're looking for, and you're looking for either the maximum or the minimum, so the peak or the valley on your topograph, and you're trying to get to that point. So if we do that, the genetic algorithm gets us right there. Even if I relax some of my conditions for my genetic algorithm, it gets me there after a few steps. For my radial basis functions, I kind of wander all over the place and I never hit the exact point, but I get pretty close. The problem is that GranSim takes about 20 minutes to 20 to 30 minutes to run a simulation. So For the default genetic algorithm, it took me almost 4,000 evaluations of that to find that optimal. The relaxed one took me 1 order of magnitude less, and finally the surrogate assisted took 21. So even though it wasn't the best, it ran the quickest. So what we did was apply a couple of ways to sort of improve the surrogate assisted, and that's what we're using now in our optimization. Okay, the other thing I have to tell you is that's for a single drug. If you want to do multiple drug optimization, we look for something known as Pareto fronts and try to find our target region for minimizing antibiotic dose and minimizing average time to sterilization. So I want to tell you in the last 5 minutes here about something interesting that we've done recently, and that is there was a clinical study recently that said that, well, you take HRZE, let's swap in this drug moxifloxacin for the ethambutol and see what happens here. Because ethambutol has been around for a while, maybe it needs some improvement. And so they said it didn't really help much. Well, we went ahead and did that same study, and what we found is, yes, you get some benefit, but that there were 2 other regimens that if you swapped out, you did much better. One, RMZE, was almost as good as HRZE, and another one, HMZE, was also good under certain conditions. So right now, Joanne and the monkeys have been testing these other regimens to see if that actually is borne out in the primate model. So finally, I want to tell you what we're thinking about relating to the translatability. So everything I've told you about, you should be questioning me because I've told you everything about a single granuloma. But I showed you very early on that monkeys and humans form multiple granuloma in the lungs, and it really only takes one bad one. So you could have 20 granuloma, 19 of them sterilize, and your body's doing a great job. One of them does a bad job, and you better treat that or that person's going to die. We do not know. We think it's location, location, location about where that initial bug lands, closer to a blood vessel, closer to an airway, closer to a lymph node. We're not really sure, but that's part of our ongoing work right now. And so what our goal is then is to be able to create a virtual human to scale to the host, and then also be able to run what we call virtual clinical trials. And so towards that goal, we're trying to build a virtual human that runs the gamut of all the different scales of interest. And so we can address questions in all these different areas of vaccine, drugs, immunotherapies, biomarker discovery, and virtual clinical trials. So to do that for TB, I want to convince you that I only need 3 compartments. I need the lungs, I need the blood, and I need the lung draining lymph nodes. And for most people, of those 90% that have latent TB in the world, those 2 billion people, these are the main compartments that TB is confined to. There's much data to show that. Of course, people get extrapulmonary TB. Children actually don't even get it in their lungs, but we're talking about the majority of people. So this is where we're starting. So what we have done is taken a non-human primate lung and digitized it XYZ coordinates, and we have that now as our background of our model. So this is a new model now. It's not GranSim anymore. We're calling this model HostSim. We've got 6 lymph nodes in the lung draining lymph nodes that are there for primates. Actually, there's like 5 in humans and 7 in monkeys, so we've got 6 to sort of balance the two. We have blood vessels in our model, and then of course we have granulomas forming over time. I will tell you that in this story, Jimmy, we are considering our lymph nodes as homogeneous, well-mixed compartments. As you know, we've done other work as lymph nodes with agent-based modeling. We don't think we need that right now. We just need them to be supplying cells to the site of infection. But later we have to add the more detail and figure out how to get them there. So what I'm showing you on the left here is our lung, and you can see I'm showing you just 2 lymph nodes now. And these are going to be the vessels showing you cells flowing from the lymph nodes to the site of infection, and you'll see the granulomas begin to start. So I don't have the key on here. I'm very sorry about that, but darker color means that more cells are flowing. And so you'll begin to see granulomas forming. You'll begin to see the lymph nodes changing color, and as dendritic cells take antigen to the lymph node, and then those cells will begin to flow. Now the lymph nodes are changing color and being able to populate these granulomas and help fight the infection. But you will also see right here, more granulomas are spreading and starting. So this example, this person is doing, or primate virtual human is doing poorly, and so you're getting the birth of many new granulomas that are seeded. And so unless we treat this individual, they will die. And so what we're able to do now is run multiple virtual hosts here, or digital twins, however you want to refer to that, and then we have a population. And so we've been using this to test drug regimens, to test vaccines, and we've published work on those areas, and you all can see that on my website. So I just want to tell you that models are very important. They're complex. I think there's a lot of tools that are needed to study these things, and considering really mechanistic models, I think, is a key to really translatable. Our tools have been the sensitivity analysis we use, numerical implementation, and how you link these compartments. And also, we don't always need other compartments running, so we do something called tunable resolution, where we fine and coarse grain our model compartments as needed as we run, and we can even automate that to happen naturally. We continuously curate our models and validate them regularly to be sure that we're staying up with the latest. We build our models middle out, and we are applying it in many areas. And we encourage, of course, collaboration. And I have a fantastic group of people at Michigan, and also Joanne Flynn at Pittsburgh, and Veronique D'Artois at CDI in New Jersey, who supplies our drug data to us. And we have a new collaboration with the Gates Foundation, with Karim Azir, and a new one also with Bree Aldridge and Sriram about drugs and TB. So thanks for your attention.
Wonderful. Thank you, Denise. You're a quick speaker. That was an incredible amount of material in a short time.
You'll have to play it back in slow motion on YouTube when you
So I don't know, anything I say at this point will be something of an anticlimax. Just to say that before we go to the discussion, we're always eager to have your feedback for suggestions to how to make these meetings better, for suggestions about how to organize the working group more effectively. We are working on a statement of goals, as people know. We'd appreciate your help. And we definitely would like to hear from our subgroups. So please, if you're a subgroup leader, be willing to report. Reinhardt, is there any business that you wanted to take before we go on to questions and discussion?
No, I don't think I'd want to interrupt the flow of that one.
Okay. Well, in that case, I think that we'll say that the business part of the meeting is done, and I will open the floor to a couple of general questions, and then in 5 minutes we have a free-for-all. So let's start out with a few not too technical questions for either of our speakers, and then in 5 minutes all the gloves can come off. I have one if nobody's going to start. I always read that humans don't have formal pumps for the lymphatic system, whereas birds do. The birds have lymph hearts. But the flow velocities you're reporting for human lymph flow are very fast. So if flow velocities for lymph are so quick, why do birds need lymph hearts to pump lymph actively?
Yeah, there's a big variety in lymphatic system physiology in the animal kingdom. So amphibians also have lymph hearts, from what I've heard. You know, it's a shame I didn't show the valves on either side of the of that pumping lymphangion, but basically for a 100 micron diameter lymphatic vessel, the valves will be about every 10 millimeters, about every millimeter, or 10 vessel diameters. So they were really just off the screen there. The lymphatic muscle cells in the walls of the vessels can contract. They do so about every 2 seconds or so. But you can also get, kind of like vein pumping. You can get external compression from external tissues pushing the fluid along. It actually does a pretty good job of moving that 5 liters a day back up to your shoulder. It's fighting gravity, obviously, in humans, not so much in birds. So I think it's, there's a big difference perhaps in the valve structure. Maybe the birds don't have valves in their lymphatic vessels. I'm not really sure, but that's a great comparative anatomy study someday to look at that for sure.
One time for one more general question before we continue the discussion. Anyone?
So my question was somewhat general. I was asking about Denise Kirschner's fantastic talk. She briefly mentioned middle out model building, so if she could just say a little bit more about that philosophy.
Yeah, thank you. So the idea is what we do is we sort of focus the model on what we really think is the compartment or the scale of major interest. And so we put our energy there first, but then of course there's peripherals. In our case, there's molecular scale details that you might blow out south, and then there's other details that you might blow out north from that scale. So in our case, we started with the granuloma, which is really what we think is the center of the universe for the TB. But then we need to think about the fact, well, there's lots of dynamics happening at the molecular scale. There's also things happening in other compartments. But we started with the most detail in the middle, the mesoscale, and then we build out north and south from that, and even east and west, adding the other organs and things on, depending on, what questions we want to ask. So for example, when we found out from the sensitivity analysis that TNF to necrosis factor, which is a cytokine, was important, we built an entire 21 equation submodel of TNF, all the bindings and the receptor ligand interactions and how they're produced and internalized, and then we can have that running inside all of our macrophages in the model at any time we want. So that would be an example of building something south from my mesoscale. But we don't necessarily need that TNF dynamics running all the time. So we have a way to sort of use this tunable resolution idea where you can turn on and off that scale model as needed, but in a very smart way based on what we learned from having that model running inside of there. So that's a general idea. I hope that was clear.
Yes, very stimulating. Thank you.
Okay, so we are at the canonical hour. I hope many people will be able to stick around and have a discussion with our two speakers. I think, as you say, sort of by accident, the talks went very well together. So good to keep everyone together. So I will now turn it over. And if people want to screen share, I can unscreen. I will unshare the screen so that you have it available. I will open it up for a general discussion. I hope we have a lively half hour of discussion.